talks in one PowerPoint, and I'm going to go in alphabetical order by last name. So you probably all know your last names and can kind of gauge where you are in the lineup. So if you could just be standing by, I would love to have, while one student is talking, have the other one, the next one in line, come up and get the mic on. So get this up. So Susanna is first. Why don't you put this on? You have to have a pocket. You can put that right in your sweater. And next up is Sophie Jou, if you want to come on up and get mic'd. Do you want to just use the arrows, or would you like to use this?
Does it have a
It has a button
pointer?
in the middle. It could be and that's just a scissors.
Hello, my name is Susanna Abdallah. I'm always first for these things because of my last name. I'm not sure if that's good or bad yet. So I'm going to start off by explaining that diatoms are so very important, and I'm sure a lot of you know that. They are very vital to ecosystems globally, and they also can tell us a lot about the environments that surround. So this transect in particular was part of the Iron Brew cruise of 2014, and this was just one of the transects. This transect is cutting across two sea surface height anomalies, and these regions that are dark blue, dark purple, are regions that is a lower sea surface height, so there's upwelling going on. And what we have is along this transect, there were surface samples collected, filtered, and then they were ready for genetic analyses. And through computational tools, we were able to group all of the sites based on community composition genetics. So what we have is these colored regions, this blue, green, and red regions, are areas where it's either greater than or equal to 80% similar. So we can see this T2 through T5, these regions right up in here are really tightly associated with one another. And to really drive that point home, we have these community compositions, and I selected the centers of each of these eddies. And so this T6, which was the offshore eddy, is heavily dominated by Rhizoslinia, and it was actually just one species that was dominating. And this T18, we have a very tiny sliver of Rhizoslinia, and a much greater distribution of all these other different groups present. So we have different things going on along the coastal region. There's a lot of factors going on because it is coastal. There's runoff and all these different mixing regimes. And for the offshore, we have really just upwelling that is the main driver of the nutrient distribution and availability for diatoms. So if you would like to see more of these community compositions, you can come to my poster either this afternoon or Wednesday morning. Correct? Yep. And that's what I got.
Just want to, Ash, you can come up.
Hi everyone, my name is Sophie Jou. I'm currently a PhD student in the MIT Hui joint program. I'm defending this September and also looking for a postdoc. So let me know what's up. Um, Before I talk about these slides, I'd just like to make a little plug for the Society for Women in Marine Science. We formed this group a few years ago to create a community of marine researchers to acknowledge and address women's issues in marine science. We're going to be having our third meeting this September 30th at Hui. And if you want more information on that, you can find me at the poster sessions or send me an email. I didn't put up there. It's s.chu@hui. Okay, so on to the science. So intertidal salt marshes are highly productive coastal ecosystems that have been previously shown to export organic carbon and nutrients, and more recently, dissolved in organic carbon and alkalinity. However, few of these studies have sufficient resolution to accurately quantify the dissolved inorganic carbon, or DIC, and alkalinity exports. So this is what I'm trying to do in these two projects, using two different approaches. And we're hoping that these results will help reduce uncertainty in current estimates of salt marsh export and evaluate the importance of salt marshes in coastal carbon cycling as marshes continue to be destroyed by anthropogenic perturbations such as land development and nutrient enrichment. Okay, so for this project, I've been using a recently developed sensor called the channelized optical sensor, which measures pH and DIC simultaneously. My field site is Sagelot Pond, which is in the Wacoit Bay estuary, about half an hour east of here. And so we measure the fluxes in and out of this tidal creek right here. And I've shown the data, the DIC in blue and pH in red from the Chanos, and the temperature, salinity, and water level in the creek measured with other in situ sensors. And you can see how the signal is fluctuating tidally. We're also interested in looking at the spring neap signal and seasonal signal. So in this project, I'm trying to do the same thing, but we don't have a sensor that measures alkalinity at high resolution. So what we've been doing is taking the in situ data that we have and making a model, multiple linear regression to match the bottle samples that we take, and then calculating a high resolution alkalinity flux, which we can then multiply with the water flux to get a high resolution modeled alkalinity concentration. I'll be presenting this stuff in my poster, so if you're interested in it, come find me. Thank you.
Hello, everyone. I am Ash Cohen. I am from the School of Marine and Atmospheric Sciences at Stony Brook University, and I am a PhD student in the Gordon T. Taylor Lab. I put up a picture of my study area. We just went there for the first time about a week ago. It's Green Lake in upstate New York, about 15 minutes outside of Syracuse, and it is a meromictic or permanently anoxic lake. So my motivation in studying this system is to look at the biogeochemical processes of lacustrine marine aggregates, which are actually 60% dry weight organic carbon, and therefore an important vehicle for sequestration if and when they sink. However, the quality and quantity of that material that is actually sequestered is determined by several competing factors, including gravitational settling, advection, and remineralization by heterotrophs. Now, in a lot of the review papers that I've read, anoxic systems are briefly touched on when we think about aggregates, but the argument is usually made that because the oxic ocean accounts for so much of the globe, they're not really important to consider. However, if you are interested in those systems, macrogates are very important to consider because at redox declines or other density discontinuity layers, there's an enhancement of shear, which, if it doesn't get too high, can actually enhance aggregation. And directly around the same place are already pre-existing aggregates of sticky purple sulfur bacteria. And then, well, at least in the case of my system and a few other anoxic systems, in the upper water column, you do have the addition of dense carbonate materials during annual whiting events. So you have all these components that would suggest that you should have a lot of aggregate formation and sinking, but you also have to consider that these aggregates can be hotspots of microbial activity. So the big question is, which phenomenon is more prevalent, and how do these competing factors actually affect the biogeochemistry of the system? And I've just put up one of the famous images from an Azam review that shows marine snow. I guess in my case, this is lake snow. That just kind of shows these competing factors. Do you have bacteria consuming this material, or does it actually enter the ocean interior? We just started studying this area, so I don't have any preliminary data to present. So that's it for now.
Okay, we're gonna have Chandler Countryman, and on deck is
Hello, my name is Chandler Countryman. I'm a first-year grad student in Adrian Bird's lab at the University of Georgia. And I'm working on developing an agent-based model for active organic carbon flux by vertically migrating zooplankton. And the question behind this project is how exactly zooplankton community structure affects carbon flux in the mesopelagic zone. And the motivation behind this is based off of suggestions and evidence that community structure does indeed affect the carbon flux. And my first example of this is this graph by Bissler et al., which shows the POC flux in two stations, Aloha, which is in the triangles, and K2, which is in the circles. And in figure A, you can see the two deployments at Aloha in the black and white triangles are very similar, whereas the two in K2, the gray and white circles, almost have a threefold difference. But then when they're normalized in figure B, the two at K2 wind up overlapping each other. And this suggests that the flux attenuation isn't due to the magnitude of the flux, but instead to processes that are occurring within the mesopelagic zone. And these processes can include food web structure, they can include particle aggregate characteristics, and they can also include any carbon demand by resident and any migratory biota. And then the second graph I have for you is this by Steinberg, which is showing the size class distributions in Aloha, which is in the top row, and K2, which is in the bottom. And when you look at the size classes, you can see that K2 comprises of larger animals than Aloha does. And in regards to the transfer efficiency that was shown by Bissler et al., it was shown that there was a higher transfer efficiency at K2 versus Aloha. And when you consider how community structure can impact this transfer efficiency, you can see that maybe the larger animals at K2 can produce larger fecal pellets, which can sink faster, which can expedite the transfer. And also, it was shown that there was a higher degree of diel migration at K2, and this can also increase the transfer efficiency by excretion at depth of particles that were consumed at the surface. And so this all leads to the base assumption behind this project, which is that much of the variability in flux attenuation arises from varying zooplankton communities and behaviors. And so what I'm working on right now is to develop a model that has individual zooplankton that have various different parameters like different swimming speeds, different migratory behaviors, different feeding strategies, and to see how they interact with particles in the water column to see how exactly different community structures affect flux. I just started this project, so I still have a lot to do, but if you would like to check out what I have so far, please come by and see my poster. Thank you.
Zachary Erickson and Laura Pillager can come up.
Hello, I'm straight. Any pronunciation is fine, as long as it's not Steven. I get that fairly often. I have been looking at a transect off the coast of Oregon that was following upwelling. They took a sharp turn here when nutrients dropped dramatically and moved back into the upwelling zone. This is temperature, showing the colder temperatures here. You can see right here that station 5 and 6, which are outside of the upwelling slope, are not very closely related to the other ones, but 4 and 7 are really closely related, which are on the edges here of the upwelling slope. I have pie charts that show that genus, the different species at each station and how that changes on my poster. I also did a correlation analysis between the distance matrix from the Bray-Curtis analysis and environmental variables, and these are the top hits here. Particular aluminum, scandium, and iron are pretty high. May be explained, they may actually just be diatoms themselves. And copper here is associated with iron uptake. Yeah, if you have any ideas of why these are so highly correlated, and dissolved iron was very poorly correlated, please come by and tell me about it. What's up?
Next up is Zachary Erickson.
So my name is Zachary Erickson, and I'm a third-year graduate student at Caltech. And here I'm showing you a mission composite of ocean color from Aquamodus. And there's a lot of things that we recognize here. There's oligotrophic centers of the subtropical gyres. There's a lot more chlorophyll around the coastal areas and in the high latitudes. And what we also see is only the top couple tens of meters of the water column. So in the bottom right insert, I'm showing you two different profiles, a surface bloom. So these are profiles of chlorophyll, and the surface bloom is going to show up pretty well in the satellite data. On the right is a deep chlorophyll maxima, which is not necessarily going to show up in a satellite picture. And as you might guess from my title, an advective mechanism for deep chlorophyll maxima in Southern Drake Passage, I'm quite interested in these things, these guys on the right. So our study area is in this red box. Although if you come by my poster, I'll be happy to speculate wildly about other areas in which it might be important. And here are just a few sample transects to show you. So this is two different transects from sea glider data. On the left, it's from a transect in late December. And on the right, it's a transect in February. And the left side of each of these is on shelf, and the right side is off shelf. And I'm showing you on the top fluorescence-derived chlorophyll, in the middle, backscatter, and on the bottom, the ratio between the two. And what I'm going to look at is these deep chlorophyll maxima that you can see around day 33, 34, 35, and transect 11. And we're going to start off thinking about them in a one-dimensional sense, so they're only dependent on what's right above them. Things like photoacclimation, subsurface phytoplankton production, stuff like that, and we ultimately find that these are not compelling arguments. And so we're going to look at two-dimensional mechanisms for their formation through advective subduction events. The black lines on here are isopycnals, and you'll notice that the deep chlorophyll maxima run straight through the same isopycnal layer as the surface chlorophyll blooms. And then, of course, we're going to think about the fact that we are in Southern Drake Passage. There's a very large ACC running through it. Everything there is going to be a fully three-dimensional field. And I'm going to go through some three-dimensional mechanisms, advective, subductive mechanisms for deep chlorophyll maxima formation using some model results. And come by my poster, and we'll talk more about it. Thanks.
Andrew Gunn can come up.
Hi everyone, I'm Laura. I'm in Bethany Jenkins' lab, and my poster is titled Characterizing the Mechanisms Underlying Southern Ocean Diatom Community Composition Shifts. So my overall goal for my research is to try and delineate the iron stress response in Southern Ocean diatoms. And here we start by trying to determine if iron or other environmental factors influence these diatom community compositions in 3 different regions of the Southern Ocean. And so here we have a cruise map of an austral summer 2013 cruise that covered the Rothera to Ross Sea transect, the Ross Sea polynya, and then in and around the ACC bloom coming out of the Ross Sea polynya. And so these are only the stations that we have sequence data for. And so I did a cluster analysis and determined that the samples clustered generally based on region. So these Rothera to Ross Sea transect samples clustered in one cluster. Then over here with these black symbols was a different cluster. We noticed that a lot of these samples included samples that were from regions with a lot of ice. So that was interesting. And then in the Ross Sea, we noticed 2 different clusters actually, which was kind of surprising to me. I thought that they would maybe all cluster together. And so looking a little further into this, so the clusters are color-coded. So here we have the green and the blue clusters from the Ross Sea. And we see that in the green cluster, Susanna renaissensis is highly abundant. In the blue cluster, there's a higher fraction of the sea ice diatom Fragilariopsis cylindrus with a lower abundance of Pseudonychia renaissensis. And then in the black cluster, with a lot of which I said before was around a lot of sea ice, we again see this high abundance of Fragilariopsis cylindrus. And then in the red cluster, there was a lot of Thalassiosira, Oestrupii, and some lower amounts of Fragilariopsis cylindrus. So we see that they are clustering differently based on the different regions. And so to delve a little deeper into this and to get straight to the punchline, iron and salinity were the main drivers of these biological patterns. So here I have a multi-dimensional scaling plot that show the samples clustered based on similarity. And so we again see those 4 clusters. And then here in these Ross Sea samples, we see that the iron is lower there. And you have to notice the scale bar, very, very low iron in the Southern Ocean, but there is a difference there that we see. And I have a similar plot for salinity that shows that in the Ross Sea, the salinity is higher, and in the other 2 regions, it's lower, indicating some sea ice melt influence. So if you want to see the rest of my poster, please come either tonight or on Wednesday morning. Thank you.
Hi, my name's Andrew Gunn. I've just started at the University of Pennsylvania with Irina Marinov last month on my PhD, but this work that I'm going to talk about in my poster, what's on my poster, is from my previous affiliation at IMAS with my old supervisor, Max. I don't have that much information in my presentation, but feel free to come and see more about how this works in the poster. But this is the final result here. Basically what we show is that we show this through idealized analytical theory, and then we validate these results with a global MIT GCM at 2.8 degree resolution. We show that diapicnyl mixing has an effect on the biogeochemistry in both of these methods, and that it would change the partitioning of carbon between the ocean and the atmosphere. And so the theory is the bulk of the poster. Basically, we follow a Lagrangian parcel around a meridional overturning cell and scalings for how dipycnal mixing affects the movement of that parcel through the overturning cell and how it inhibits the amount of nutrients available in the surface ocean, has effect on the amount of carbon that can be stored within the deep ocean. And so we have the result here that we try different dipycnal mixing values in the global circulation model and also in the theory, and show that there's a good agreement between the two that as dipycnal mixing gets stronger and the overturning enhances, that there's going to be less DIC within the ocean, and that in turn would increase the atmospheric value if it was a simple partitioning between the two. And that's all I want to say today. Thanks.
I got one. Thanks. Oh, thanks. Hi, my name is Evan Howard. I'm a graduate student in the MIT Hui Joint Program. I'm presenting a poster about using noble gases to compare gas exchange parameterizations in a shallow salt marsh pond. My co-authors are my advisor, Rachel Stanley, and Inka Forbrich. And so I guess what I'd like to talk about is the salt marsh pond. Coastal environments are really productive, including salt marshes. And they have big diel fluxes of oxygen and CO2 and other metabolically active gases. So a common technique for measuring metabolism in these environments is to measure the fluxes of these gases. But that means you need a good estimate of gas exchange. So we use noble gases, which are an in situ natural abundance tracer that's sensitive to a bunch of physical processes, including gas exchange and bubble processes, to compare these gas exchange parameterizations. And for the most part, we find that they all fit pretty well, but you can see my poster for that. One of the things we find is that there's some undersaturations in the, sorry, you can't really see the title there. This is neon saturation in percent, so 2.5% supersaturated, 2.5% undersaturated relative to atmosphere, argon, krypton. And there's some undersaturations in the more bubble-sensitive gases that are plausibly explained by ebullition. And anecdotally, we see lots of bubbles rising from the sediments during daylight, which might indicate ebullition. And we find that the ebullition rate that we estimate from the noble gases, which here is the black dashed line, tends to track the oxygen saturation state, which is the blue line here. So plausibly, this evolution has to do with generation of photosynthetic oxygen in the sediments. And if you were to wildly extrapolate, the rate that we observe here could account for 10 to 20% of total net oxygen efflux during the day. And that's a flux of photosynthetic oxygen that would be missed by gas exchange parameterizations that don't explicitly include things like these bubble fluxes. Thanks.
Hi, my name is Grace Kim, and I'm a PhD student at Johns Hopkins University. And my poster is all about modeling changes in ocean color. So I'm showing you two images here from the Arctic Ocean. And you can see the clear difference in color. And you can imagine that these changes in color and the differences in color are telling you something about the way light is attenuated beneath the surface of the ocean. So in this image, light will hit the surface of the ocean, and as it goes deeper into the ocean, it's going to be attenuated by water itself and some phytoplankton. But in this image, which is closer to shore and on the shelf, there's going to be light attenuation by water and phytoplankton, and also colored dissolved organic matter, which is giving the water this brownish color. And so if we think about modeling this in an Earth system model, we want to be able to capture these differences in ocean color because it's going to change the primary productivity and the heating in the model. And current IPCC class models do not include the contribution by colored dissolved organic matter. So what I did is I added in colored dissolved organic matter into the Earth system model, and what we found is that there were shallower attenuation depths, so that's a shrinking euphotic zone. We also found more extreme sea surface temperatures, so warmer and colder temperatures from the control run, and more ice in the northern hemisphere. And we also found that there's less total biomass and less carbon export out of the surface ocean. So if you want to know more about modeling ocean color, stop by my poster. Thanks.
Andrew Margolin and Sarah Rosengart can come on up. This is Taiwan Kim. Yep. Pronouncing your name. Is this? Nope, you're fine with that. Do you want to stick it in your pocket or something? We're just doing the opposite. Let's get you advanced here.
Sorry, I didn't put my title of the poster. So the title is Parmo LTER, Climate Biogeochemical Coupling in an Antarctic Coastal Ecosystem. So I'm Helen Kim, the third-year graduate student with Dr. Hugh Duclos at the Mount Doherty Earth Observatory at Columbia University. And my research has been largely involved with Parmo LTER program, where we have observed key ecological and biogeochemical variables along the Western Antarctic Peninsula since 1991. So we have a core working hypothesis about why we study this region, that sea ice is a mediatory physical forcing factor for shaping ecosystem dynamics and carbon and biogeochemical cycling in the Antarctic ecosystem. So to test our working hypothesis, we have operated annual LTER cruise with RV LM Gould every January, and also deployed to Parmo Station for performing biweekly zodiac sampling during austral growing seasons to measure regular monitoring and biogeochemical ecosystem variables. So my research question is about how ecosystem and biogeochemical functions interact with large-scale climate variability and upper ocean dynamics. So this is just a really short summary of all the work I've done. So I worked on analysis of observations, including dissolved inorganic nutrients and phytoplankton and heterotrophic bacteria from Parmo Station for the last 20 years to look at interannual and seasonal variability of those variables and how they are shaped and predicted by large-scale climate variability, things like sudden annular mode or ENSO and its associated physical forcing mechanisms such as autocomb stratification and sea ice. And so some of the major findings were climate and physical forcing mechanisms indeed influence the interannual variability of seasonal nutrient drawdown and bloom dynamics. And also heterotrophic bacteria are closely coupled with phytoplankton variability because there is not much of external source of dissolved organic carbon for heterotrophic bacteria in Antarctic waters. And nowadays, I'm developing nonlinear models which really targets to distinguish a true causality between two time series variables from simple correlation, which is called convergent cause mapping. So before employing this method to Parmo LTER time series, I'm testing with the Bermuda ecosystem case because there is really well-known and well-developed ecosystem model is available along with observational data from Bermuda Atlantic time series. So with that, I could test sensitive tests and to test the best model to really understand if this model really works for the time series of our interest. So my ultimate goal is to employ this method to Parmo LTER time series to revisit the strength of mutual causal relationships and the phytoplankton and bacterial coupling. And also I want to look at the carbon flow and its interaction with upper ocean physical forcing. So please come by my poster if you want to talk more about this. Thank you.
All right, so my name's Andrew Margolin. I'm a final year PhD student, and so I'll be finishing in May 2017. I'm working with Dennis Hansel at Rasmus at the University of Miami. And the poster I'll be presenting here is on net removal of dissolved organic carbon in the anoxic waters of the Black Sea, which was recently published in Marine Chemistry. And this is a map of the Black Sea showing chlorophyll, and then you can see the stations marked in red there. So if you want to learn more about the DOC in the Black Sea, you should stop by the poster. And then more broadly, my dissertation topic is on the environmental impacts on carbon biogeochemistry in marginal seas. And so this is just a map of the world, and in color you can see the ocean's margins between 0 and 1,200 meters depth, which cover about 10% of the Earth's ocean, or about 7% of the Earth's surface. And if you look into marginal seas, such as like the Caribbean or the Mediterranean, you fill in that notable area becomes even larger. So in order to understand ocean carbon biogeochemistry, it's important to also understand the biogeochemistry in the marginal seas. And so in addition to my poster on DOC in the Black Sea, I will also be, for my dissertation, another chapter will be on chromophoric dissolved organic matter in the Black Sea, or CDOM, hoping to gain a better understanding of the river input of terrigenous matter into the Black Sea. And then I'm also looking at dissolved organic carbon and CO2 in the deep Gulf of Mexico and Caribbean below the sill depths. And just as a bonus, last summer I participated in the US GeoTrace's Arctic expedition, sampling for DOC and measuring the CO2 system. And so if you stop by my poster, ask me questions about any of these or bump into me in the halls. And also, I'm on Twitter, so if you guys don't get a chance to bump into me, you can send me a tweet. And I also want to acknowledge my support from the NSF and my home institute. Thank you.
It's done? Oh, it's done. Cool. Oh, cool. Hi, I'm Sarah. I'm probably, most likely, in my last few months here at Hui in the Marine Chemistry Department. About half of my thesis is on figuring out the factors that enhance or inhibit the efficiency of organic carbon export via particles. And so I hope that by the end of these 2-ish minutes, I'll convince you to be interested in a unique method that I'm using to characterize the composition of particulate organic carbon in the ocean. And I've applied this method to particles collected from a calcite-rich region of the Southern Ocean called the Great Calcite Belt. So I'm showing that in the top panel, those are MODIS-derived images with lighter colors being elevated concentrations of particulate inorganic carbon. And so there's a lot known. We know a lot about the bulk composition of the particles we collected across the Atlantic and Indian sectors of this feature. We know a lot about the bulk carbon export and transfer features across stations here. For example, these profiles that I'm showing below. But less is known about how POC quality influences the differences in the carbon export regimes that we observe. So to get at quality, I subjected these samples to ramped oxidation, which separates organic matter according to its thermal stability. So I'm showing a schematic of this figure on the left that my friend created. So in an oven like this, you can subject organic matter to a controlled ramp from room temperature to 800 degrees Celsius, and monitor the amount of carbon dioxide that's created in the oxidation process throughout the temperature ramp, and plot it in the form of thermograms like this. And so what I'm showing here is a spread in the thermal stabilities you would expect in what I call marine end members. So like pure phytoplankton cultures that I grew in lab in green versus almost pure particulate inorganic carbon standards or sediments in the gray and the black. And you can couple these thermograms to a vacuum line and collect
carbon dioxide at specific temperature intervals and analyze the isotopic composition of the gas throughout the temperature band. So that's what I'm showing in this thermogram on the right as the height of the bars. The height is the delta, the stable carbon isotope signature. And you can use these isotopes to diagnose the identity of components that have different thermal stabilities in the thermogram. And also figure out different methodological caveats to this method. For example, how matrix effects in a sample would change the apparent thermal stabilities that you observe in a thermogram. So if you come to my poster, I'll show you how I use isotopes to get at those matrix effects, and also ultimately interpret changes in organic carbon composition throughout the water column via real sample thermograms, which I'm showing on the left. So I have a lot more of these to show if you visit my poster. Thank you. Oh, no, that's sorry.
Hello, everyone. My name is Jens Terha. I'm a PhD student in Paris at the IPSL, working with James Orr and Laurent Bob. And I'm using a global circulation model with, here I think, yeah, it's half a degree resolution and I'm working on the Arctic. So what I was looking at was the anthropogenic carbon, and I looked at the budget and wanted to know where the anthropogenic carbon gets into the Arctic and where it goes out and how much is stored every year. So here you see in units teragram per carbon per year. And for me, I was quite surprised that most of the carbon doesn't come in by the atmosphere, but it comes in via lateral transport. So from the net input, it's 75% comes in by lateral transport. And for my PhD, for my last 2 years, I will look at the effects of rivers. So if anyone knows something or can help me to quantify the amount of anthropogenic carbon that goes into the Arctic Ocean by rivers, I would be quite happy about any help. And just to give another view on the carbon inventory, or the anthropogenic carbon, so we see that it's mainly stored where the ocean is deep, what we could expect quite easily. And if you calculate, or I calculated the cumulative flux, I was another time a little bit surprised, but it was suggested in some studies, but still argued that there's outgassing of anthropogenic carbon, so not natural, but really anthropogenic carbon. It's one of the only spots in the world where you can see that. And then I calculated the difference, and it shows quite nicely where we have not much input of carbon, but still storage, or where there's a lot of carbon that goes in, but it's not stored and transported away. So red regions show regions where carbon flows in, and blue where carbon flows out. And here I calculated the anthropogenic carbon inventory to be 2.7 petagram in 2007, and to see if that fits quite well to observations, what we actually found out. And if you want to know more, you can see my poster there. I compared the CFC and observations with the CFC and the model. And I think it's a little bit better than the models that Scott Downey showed this morning. So I was quite happy about that. Thank you.
We're in the rare position of ending exactly on time, which is, it took the students to show us how to do that. Just a couple of things. So the shuttle's going to meet you right outside where it dropped you off, right out in front of Redfield. Oh, they moved it. Okay. All right. Too much going on. Okay, so out by the tent. That'll get you back to Clark 507, where we'll have a poster session with more snacks and drinks. When you get there, if you haven't hung your poster already, please do so, and take advantage of space. Like, try to space them so that we can fit them all. It's like going to the grocery store and trying to park cars, and people park this far apart so nobody can park. And try to just maximize the use of the space so we can make sure all posters can go up and stay up for the entire meeting. I just want to do one more thing for Mary. I hope this is the last thing I have to remember. I would love to get an estimate of who's planning to come to the film festival tomorrow night so that we can plan accordingly. There'll be snacks for that too. We're big on snacks here, if you haven't figured that out already. That's a pretty poor showing. Come on. Okay. All right, is that good, Mary? All right. I want to thank everybody who spoke again today for great sessions and thank our session planners. And we'll see you up at Clark 507.